the different methods which are used for determination of antibodies and antigens, we will discuss another very precise and important technique, and that is immunofluorescent microscopy, IF, which is briefly known as IF, immunofluorescent microscopy. And in this immunofluorescent microscopy, we use antibodies which are conjugated to fluorescent marker. You see, to determine the antibodies, we have to use the marker. They have to be marker. It can be a conjugated protein, it can be a conjugated enzyme, or some other marker which impart the color. Now, in this method, we use the antibodies which are conjugated to fluorescent marker to locate the specific structure on specimen. Now, this is very helpful to determine the structural specificity and the structure which are there on the specimens. Now, it allows the specific structure to be visualized by illuminating them with ultraviolet light. So you see, previously we use the spectrophotometry or other things to determine the concentration, but here we can use the illuminating them with ultraviolet light when UV light, as a matter of fact, reacts with the fluorescent antibodies. Now, fluorescein and rhodamine, these are the two compounds which are usually labeled and used for alternative markers which are available. So we can, with the radioactive as well as the fluorescein and rhodamine, they are also the labels used for determining the IF. Now, fluorescein produces a green fluorescence, and rhodamine, it produces a red fluorescence. So there are fluorescein and rhodamine. One produces green color, another one produces a red color. Now, microscopes which are equipped to carry out IF have dual sources of light. So they both have dual sources of light to determine the concentration of these, and this white light allows the operator to view the specimen before illuminating with ultraviolet to look for specific fluorescein. That if there is a specific fluorescein, that is whether it is a red or green. So with the white light, first it is used. The technique is used for looking surface marker on eukaryotic cells. That is, on the eukaryotic cells, what are the different surface marker present? We can use IF to determine that. And another use is as a whole cell staining technique in bacteriology. That is, whole cell can be stained and we can determine the structure of the whole cell while studying the bacteriology. Now, endocytosis, that is the uptake of membrane-bound particle by the cells. You see the membranes which are present around the cells, they take up or uptake certain particles, and that can also be determined by the IF technique. And these studies include studies on whole mammalian cells, that what whole mammalian cells behavior is during endocytosis, the migration of the compounds or migration of different chemicals, whatever. Endocytosis, that is the inclusion of particles by the membrane. And then also we can study the fate of labeled receptors, that how the labeled receptor on the surface of the cell can behave or what is their fate. Now another thing is the capping. That is, the bound receptor in the cell membranes frequently migrate to one end of the cell prior to being endocytosed. That is, before being endocytosed, before being taken up by the cell membrane, we can study that how these bound receptor, they migrate, how their migration, how takes place to one end of the cell from one place to another, and that gives us an insight look toward the behavior of the cell, and these can be studied by the IF technology. So you see, now we have at hand different techniques which are used or which can be used to studying the different behavior of the cell, the concentration of the antibodies which are present, antigens which are present, and also the behavior of the molecule on a whole cell, that how the whole cell is behaving during the presence of certain particles.